I'm very honored to introduce Dr. David Smith. Dr. Smith is an infectious disease specialist and a translational researcher with a focus on turning lab discoveries into human health. Dr. Smith has published over 360 scientific publications. And in 2022 and 2023, Dr. Smith was recognized by Clarivate as being in the top 1% of researchers in the world by scientific citations. Since the pandemic started in 2020, Dr. Smith has been engaged in an international effort to find safe and effective treatments, including being the international protocol co-chair for the ACTIV2 treatment study, which is a part of U.S. government's response to COVID-19. In 2023, Dr. Smith became the Assistant Vice Chancellor of Clinical and Translational Research, where he leads a team of scientists to tackle the difficult biomedical challenges. So having his breadth of expertise allows him to better understand and overcome scientific and medical roadblocks. A good example of this is his recent work that showed how ChatGPT was better and more empathetic at answering patient queries than physicians, and how these new AI tools need to improve how a patient feels, functions, or survives to be most useful. Welcome, Dr. Smith.
Thanks. Everybody can hear me okay? All right. So we're going to talk a little bit about death today. Hope it doesn't make anybody uncomfortable, but it's all good. It's part of life. So as if the way one died mattered. Here's my disclosures consult for a few biotech companies, but the objective of the talk today, in full disclosure, is to entice you to think about research questions and how you might want to partner with us. Okay, that's my commercial. Whole thing's a commercial. Okay, some quotes to get us started is my chivalric fool, as if the way one fell down mattered. When the fall is all there is, it matters. Okay, and then we're going to talk a little bit about the end. So to start with that, put your thinking hats on. I want to ask you a question. You don't have to give me the answer to that question, but I want you to think about it. Would you be willing to participate in a research study at the end of your life if you had a terminal illness and only had 6 months to live? It was a very hard question for NIH reviewers to get their head wrapped around. But we asked a whole bunch of people this question, both people who had HIV and people who didn't have HIV. Would you be willing to participate in research at the end of your life? 80 to 90% of people said, yeah, I would be interested in that. What about organ donation? Pretty much very similar. How about if you participated in this study and it meant that you had to give up a few days of your life? Hard question, right? A third of people said, I don't want to give up any days. But some people said, I would be willing to give up a month, almost a month. And then some people said, I'd be willing to give up more than a month. In fact, people with HIVs, a third of them said, for the chance of participating in a study at the end of my life that meant nothing to my care, I'd be willing to give up a month of my life. That's a lot. And of course, they'd be willing to do injections and blood draws. We asked them a bunch of those questions, but this was really sort of eye-opening to us. Okay, I want to change gears a little bit, but I want you to percolate that in the back of your mind as we go forward, okay? So I'm a virologist, even though I sometimes dabble on ChatGPT, which is a little embarrassing. That's probably the biggest study that was referenced last year of anything that I've done. A little bit nuts. But my heart and soul is around HIV infection. I want to cure it in my lifetime. I'm an infectious disease doctor, gay man growing up in the 80s and 90s, and this is what I want to do with my career. But to cure HIV, we really need to know where the reservoirs are and how we can clear them out. And in reality, we do a lot of testing in blood because it's relatively easy to get. But that isn't the reservoir, or it isn't the most of the reservoir. It's just a small glimpse of it. And even that glimpse of it isn't the same as the reservoir in all the other places. In the anatomic and the solid anatomic sites with solid tissues are really important. And HIV just plays a hide and seek with the immune system in those sites. And they can really be a source of viral rebound once antiretroviral therapy is stopped. Okay. And finding these reservoirs are hard. In living people, it's hard to biopsy the spleen. It's hard to look at the liver. It's hard to look at the heart. So we really need to get in there and do it, but you can't really do it in live people. And autopsy studies have been done ever since the beginning of the AIDS epidemic, but they often are done late. So there's very poor characterization both before somebody passes away and also done fast enough to really get the tissues to be able to see what's going on in viable tissues later after death. So we started this project called Last Gift, and it is where people who are at their in the last 6 months of their life or less enroll in our study, and it's observational. We just watch them. We take a little bit of blood, we take a little bit of general secretion. Sometimes we get a biopsy every now and then, or if they get a biopsy for clinical purposes, we get that as well. But for the most part, we just watch them, and then when they pass away, we do a rapid autopsy. And here's two of our very early Last Gift participants, 1 and 5, and they wanted to be very much up in front. They want their names to be known, Tony and Michael, as participating in this study. Interestingly, both of these gentlemen died of ALS. But a big component of why they wanted to do this study was to give back. And there's a really nice video online if you want to look at it on Last Gift, but also they're making a documentary on this gentleman's last few moments of life. Okay, so here's our potential solution to look for these reservoirs. We take altruistic people with HIV with a terminal illness, cancer, cardiovascular disease, ALS. We follow them, collect some data and some blood, and perform a rapid autopsy. And this is not necessarily new. It is new for HIV, but not new for cancer. In cancer, they call these warm autopsies. I don't particularly like that term, but rapid. And we collect all these tissues within 6 hours of death. We've actually gotten quite good now and can really get them within 3 to 4 hours, all the tissues collected within death. And that really gives us some very viable tissue to see what's going on in these different environments. Preserves the RNA and the proteins, etc. And this is our group age breakdown. We can see that we have a few 80-year-olds, 70 to 80, etc. Blue is the men, but we have a lot of people who are less than 50 years old. We're only funded to get 5 autopsies a year, with one caveat, which I'll talk about in a second. And we actually have more people who want to join our study that we have spots for. So we have been sort of selecting for people in the lower age ranges lately to get a different look at tissues. However, we would like to get an aging grant to look more at the older group as well. Okay. Other things that we collect are the comorbidities, and here is just we have people with seizures. This is just high blood pressure, which is common in this age group in general. We have a lot of people with cancer. We have people with hepatitis C, diabetes, etc. So what we can think of is, what are people with HIV have a lot of comorbidities. In fact, that's oftentimes why people with HIV do worse than people without HIV. And we're starting to think, how do we study it in the context of all the tissues that we're collecting as well? And the other one is, these people are not dying of HIV. They're dying of these comorbidities. And specifically around cancer, people with HIV have a higher risk of cancer, not just AIDS-related cancers, but all cancers, specifically breast cancer, lung cancer, brain cancer, liver cancer. So really trying to figure that out might be a good cohort here. Okay, and then we work really hard to collect antemortem data. So before death, we try to get as much history. This is actually LGO1 that you saw with his partner Blake at the beginning. And then we collect as much as CD4 count, viral load, what therapies they were on, etc. And this gentleman actually stopped when he got his terminal diagnosis, he stopped his therapy. That gave us a very interesting look, right? So we could then see the virus as it suppressed DNA. Once it stopped the therapy, then our RNA went up and we could see where it went throughout the body. And I'll talk more about that story in a second. So very interesting. We didn't ask them to do it. Just some people like, I don't want to take my medicine anymore. Okay, we can watch you do that. We have some people who are suppressed all the way through. You can see where there's some blips, etc., and we can collect those and really understand what's going on. And sometimes we actually have, they have been in other studies where there's blood associated, where we can actually go back, or tissues that are associated, that we can then figure out what their longitudinal look at their virus and their reservoirs. And there's some people who don't take their medicine very well, who remain lots of viral loads flipping around that we have collection over years. Other antemortem data that we have, we're very focused on neuropsychiatric data, and HIV-associated neurodysfunction is very common. And part of this project is funded by the National Institute of Mental Health. So they're very interested in the study in brain. I'm going to talk a lot about brain data today because they paid for it. But we get lots of antemortem data on how well they're doing with their neurocognition. And what we found is that most people actually have some mild to moderate neurocognitive dysfunction very early on, which is very consistent with the literature with just people with HIV who are very well suppressed. Other things that we collect, substance use. So people use substances. So tobacco, stimulants, marijuana, cocaine, amphetamine. Patient the other day told me his favorite weed was cocaine. Okay. And here is where we can also collect what those different types are and how, when they use it right before they die. And then that helps us when we go back, and then we can look in the tissues, which I'll show you in a second. But then they pass away. So all that was our antemortem data. They pass away, and this is our team. So we get a call. Usually we know ahead of time that someone is deteriorating. And usually, so then we start the watch basically, and we start labeling tubes and we get everything ready. And when it happens, this is the morgue. And then this is what it's our little moment of silence. We read this person and have a moment of silence. That's what we're doing here. And then we have a diener who does the autopsy basically and gives us the tissues. And those are the tissues in these little buckets there. And then these are the cutters. I think I'm in the middle over there. And we cut them up, put them in tubes, and then we flash freeze them. That's over here. And then we also have some RNA later, and we do some other ways of preserving them. And then if we're interested in the single cell RNA-seq study, that's also done in this back room. So we get that tissue right in the morgue as fresh as possible. Here, once we have those tissues, we can go back and measure morphine equivalents, for example, in all these different tissues. These are all the last gift participants that we've looked at this in and really see what the penetration is. We can also do antiretroviral therapy levels. You can see in the very low corner, that's a central nervous system, very little antiretroviral penetration, while the GI tract, which is in green, and the GU tract is in yellow, much higher. But this is lots of variation. You can see little dots in different people. And then we also do a lot of cytokine studies, so looking at inflammations within the tissues, in the blood, and before they die. There's also this syndrome called terminal illness syndrome. So some people, not everybody, but about 20%, it looks like, have this very big inflammatory stuff that happens right before they die. So their TNF-alpha goes up, IL-6 goes up, and you can really see them progressing quite quickly to death, but in that inflammatory state. So we separate those out. We don't necessarily look at them. We look at those differently within the reservoir than we do with the other group that just stays the same throughout the whole time. And using these cytokines, we can really figure that out. Other things that we can do is look at the microbiome. When people talk about the microbiome, they're usually talking about the rectal microbiome. But in fact, you have a microbiome that is different all across your GI tract. So really for the first time now we can really see what that difference is in each of those tissues, from the duodenum to the ileum to the jejunum, etc. And it looks different, by the way. That's the take home, but we're still analyzing what that means in terms of the reservoir. Most HIV reservoir is actually held in the gut, and that gut is very closely associated with these bacteria and fungi, etc. So trying to figure out what those connections are is something we are working on a lot. Okay. Any questions about that? Because we're about to talk about the reservoir itself.
Can you comment more on the associations between the microbiome and the reservoir? Like what have you found so far? What are you looking for? What would be a hypothesis there?
Yeah. So it turns out that what we suspect, that Fusi-bacteria is very much associated with an inflammatory gut, and especially if it's in the higher GI tract. You get a lot in colon, everybody has it in their colon, but it's very rare in the jejunum and the ileum. And if we do see it there, it turns out that it looks to be associated with a higher HIV DNA load there. So that's just one thing that we're looking at, but there's a whole bunch of, I don't want to get too far above my skis, but it does seem to be very inflammatory in that setting. And that inflammation is associated with higher reservoirs. The other thing is the stool microbiome is horrible at figuring out what is actually going on in the reservoir. It's relatively uniform in people versus the higher up. And yet that's what everybody looks at because it's easy, right? It's the same thing for blood. It's easy to get. Most people poop every day, and there you go. Okay, viral reservoirs. So here is total HIV DNA in the various different compartments: brain, gut, GU tract, lymphoid tissues, and others like adipose tissue and lymphoid, lung, etc. And you can just see that it's sort of all over the place depending on who they are. So there's variability. That's actually not a bad thing, which we'll talk about in a second. But the general patterns relatively track. Like the GI tract has a lot of HIV DNA, as everybody said. The DNA is quite a bit lower, but even within the brain has low levels of DNA, but it's different across the different groups. Like parietal cortex is almost always low, while the spinal cord is always about in the middle. But if you look at the basal ganglia or the hippocampus, it always tends to be a little higher. And then the GU tract, the testes are relatively low, while the prostate, which has actually a lot more blood in it, is higher. But the variability is interesting because every time that we start associating with inflammation, you can see that inflammation really does predict how much HIV DNA is in there. We can also look at activity. So one of the nice things is the DNA, well, just to back up, the DNA, we've known this for a long time because autopsy studies have shown this, because DNA is stable. You can wait 24 hours to do that autopsy and you'll still be okay with measuring DNA, but not for HIV RNA. So we're just now really sort of plotting out what does HIV RNA. So these are people who are fully suppressed, but they're still popping off transcripts of HIV RNA, and then we can pick that up and measure it in different tissues. And that's what's the sample on the bottom, and then the different participants are on top. So we're then seeing that the gut basically, actually this is all brain. So in the brain, the basal ganglia is always popping off HIV RNA.
You had showed earlier that you don't have great ART penetration in the
Exactly.
CNS, so it's a lot of this active replication.
We don't see forward evolution when we do phylogenetics, which I'll talk about in a second. So I think there's probably enough ART in there to keep it from going forward, but we are seeing it hopping off. And we also don't know if this is replication competent as well. Anyway. But going back to the point about neurocognitive dysfunction, maybe that's the reason why. Somebody having a virus popping off in their brain can't be a good idea, right? And maybe if we don't have good ART penetration too well in there, maybe there is something that we need to do better to protect people's brains when they have HIV. To get to that point, what about the reservoir in the brain? Well, this is a study we helped with recently with the Duke group, with Gladstone and his group, and they very interested in microglia. So we had fresh brain, and they had a way to pull out the microglia and the CD4 T cells in the brain. And then that's up here. So CD4 on this side and microglia on that side. And they had an outgrowth assay, and we gave them 4 donors. And the outgrowth assay, the only thing that grew out from only the microglia had virus that was productive in that group. And it was mostly entropic virus, so macrophage R5 virus. So that's pretty cool. There was a long debate about whether or not microglia were productively effective and could actually be a reservoir. And this kind of said, yeah, they can be, which is kind of bad news, right? But there it is. And the only way we would have got that is because we actually had the fresh tissue. Oh, and then that little graph is where they put an LRA latency reactivating agent, and was able to pop that virus out, keep growing from the microglia. Okay, so now we're kind of focused on the brain a little bit. So this is all about sequencing. So everything I did before was just like measuring quantities and outgrowth. So HIV DNA levels, you can see differences across different parts of the brain. Frontal cortex, basal ganglia is always nice and high. And then we look at viral diversity and how that can correlate. So we do our sequencing, and we can see some populations are quite big, and then other populations are quite narrow, even though they might have a lot of HIV DNA in them. We can also look at co-receptor usage just by sequencing the envelope. And we see across all the tissues that, boy, the gut really likes its R5 virus, which we've known for a while. Well, the brain also likes its R5 virus, which we now know is probably associated with the microglia that's there. But other parts like the lymph nodes, the spleen, and the liver, if you're going to find X4 virus, it's almost always there. But you can now start to see that. And then we can do these things called phylogenetic, where we do the sequencing, and then we look at, use some Bayesian techniques to model across how the virus gets to where it's going. Okay, so what's interesting about this is these Bayesian techniques can start to time when the virus gets to certain places based on its evolutionary history. And what was really interesting is that the people who stopped their therapy, right, and we were able to measure the virus that was in the DNA, the virus that came out, that rebounded and then evolved during that rebound time, gave us a clock. So once we had the clock, we could start timing where those viruses would have popped up, and we could start seeing some clusters. So here are brain clusters that I put into the arrows. And here's our Bayesian distribution, and we can see that some were very old before that person probably started their therapy, and then these other ones happened afterwards, basically. So you can start to see how repopulation happens in different tissues when rebound actually occurs. And then you can start making maps. So these are people who are on and off therapy. So even people who are on therapy and virally suppressed, especially if we have sequences that predated before they started therapy, we can start making maps and figuring out where the virus goes. And what's interesting is blood is the highway. And it's the highway for both people who stop therapy, that's this one, it repopulates lots of tissues, PBMC, that then goes to the brain, and then to the spinal cord, to the gut. But it also goes into people who are on therapy. We can actually still see migration from the blood into the lymph node, into the brain. Also, the brain tends to exchange. So a virus that's in the basal ganglia will go to the parietal cortex. But what was really interesting to us is that lots of migration happens in the gut while somebody's on therapy. And we're pretty sure these are all HIV DNA because they're suppressed, is there's migration of those T cells that are infected. Go ahead.
Excuse me, just a quick question. Do you know if this kind of transmission is carried on by cell-to-cell contact or by free viruses in the blood?
But we don't, so the people who are, so this one, it's all free virus pretty much. So they have high amounts of viremia, and we can follow that it is the rebounding virus that then goes in. So for people who have stopped their therapy, it's all HIV RNA, probably cell-free virus that is going there. In the people who remain suppressed, we're pretty sure that it is cells that are moving. HIV infected T cells that are moving to another site. So I want you to remember that because we're going to talk about that in a second.
Can you predict from the right graph where the virus is coming from in the rebound?
Yes. So in this graph, it's almost always the PBMC. Not always, but almost always. Sometimes, well, actually, that's not true. So in the blood gets populated by blood. The gut gets populated by the gut. And sometimes that gut virus spills over to the PBMCs in the blood. It actually takes a little while.
What is that green arrow from the spinal cord to the blood?
To the colon. So in this one participant, we saw a nice little connection from blood plasma hit the spinal cord and the colon, probably at the same time. Yeah. And then we have this hippocampus that has its own population, and it's HIV RNA, and it wasn't seeded by the blood, it just made its own. So that was pretty interesting.
Do you think it had its own reservoir? And
Yes.
when the drugs were removed,
It just made its own, and it did not get seeded by the blood. It just made its own compartmentalized virus. And we see this actually not infrequently in the brain, not just the hippocampus, the basal ganglia sometimes will do it, the frontal cortex will do it, just make its own virus. Okay, but I want you to hold on to that notion that HIV infected T cells move, okay? Because we're gonna talk about that in a second. Before that, we're gonna talk a little bit about BNAbs because everybody's hot on BNAbs, except me maybe, but they're used a lot and people are really focused on them in the cure. And even for therapy, Gilead has a whole program around broadly neutralizing antibodies. And it might be really good because they can last a really long time. So it can be used as a therapy, maybe one infusion, you won't need another one for 3 months or even 6 months. So maybe that's really cool. But the problem is, at least for the antibodies that we have now, is that the virus is really good sometimes to get around those. Even though we call them broadly neutralizing antibodies, the virus finds a way. And the susceptibility is not always the same. So what we see in this graph in the top one is a broadly neutralizing antibody called PG9. And it looks like this person's susceptibility in all these tissues that we measured was pretty good to PG9, except in the brain. So if you were to take this person's blood and you're like, okay, is this therapy going to work for them? So we sequence the virus and we find, oh yeah, PG9 looks great for the virus we find in the blood, right? And we were to give it to them, it actually turns out it wouldn't work very well for that's in the brain. So using our rapid autopsy, we can actually start to look at what the susceptibility of the virus and all these different reservoirs are across different tissues. And it turns out the brain compartmentalizes virus and it grows there, and we'll have different susceptibilities to other monoclonal antibodies as well, like that other PGD.
And that's consistent between donors. It's always the brain that is.
No, not always that it's not susceptible, but across donors, it is often a different population. Gut also happens to be a common place to see a different population with different susceptibilities. Yeah.
What do you think about these treatment interruption studies then? Because different tissues, the reactivation of the virus might be happening at a different point of time, and just the measurements that we do, we might not be able to figure out if it's getting reactivated in the brain earlier, and waiting unnecessarily long on that interruption.
So the current treatment, great question. So the treatment interruption studies that we do now are great. They tell us a whole lot of stuff, but it is almost always the blood. So we see that the virus come back in the blood, it comes back relatively quickly, and we put the therapy back on for those people. And that means that we're missing the late reservoirs coming up. And then I'll just go back to that. These are different places in the body that has erupted these. These are actually CNS. So there are different places that interrupt the virus rebounds, and then they populate those in different times.
So you think that the virus gets reactivated and testing it in blood is probably the earliest time point when we test it, and other tissues it reactivates later?
Yeah. Pretty clear. Well, the colon, the whole gut also, and the blood, they are there first, right off the bat. But then people get placed on their therapy. So we're never gonna see, which we shouldn't see. We should put them back on therapy. Those patients need their therapy and, to be safe, they need to go back on. But this, at least, this cohort allows us to look longer. So then we can start to see where these reservoirs start to reactivate. Adipose tissue is very interesting. It pops up late, but it can be quite active when it does get going. Okay. Any other questions about the reservoir?
ENOPs go into everywhere, CNS.
Most do. I mean, they're not great to get into the CNS. The blood-brain barrier is a real thing. Also, the blood testes barrier is also real. So they don't get in all that great there. But they are making these neutralizing antibodies, at least for commercial purposes, to be able to get into those tissues. Okay. Other things we're looking at is the immune environment. So we talked about the viral environment, my shtick, but we're also looking at T cell receptors, and you can track those over time and space and place within a body, and you can look and see what clonotypes there are in PBMCs in different people. This is just show that that's possible and what the proportions look like. And you can then map them to different tissues. So you can take each of those tissues, sequence the TCRs in each of those places, and then start to look at it. You can also see who shares, like does the PBMC share with the colon versus sharing with the colon with the jejunum. So you can start to get a nice map of where all your TCRs are and how they're shared across tissue. Well, told you we could do the same thing for the virus and how it migrates across the body, right? And what tissues are then sharing or moving of that virus around. And then you can correlate those two. It turns out they're very correlated. That TCRs that are shared are also oftentimes sharing the HIV. So it goes back to that point that I made before. HIV infected T cells are moving, right? And if you look at the TCR receptors and you're sequencing them, you can actually see, boy, they are something about the jejunum and the colon really like to share T cells. And the spleen really likes to share T cells with lymph nodes and the liver. And these are HIV infected cells. Okay. Any question about that?
So you think the migration is TCR driven, like specificity?
I don't know. I just know that they are. I think so. If I had to put a nickel on it. But I don't know. Okay. All right. Now I'm just going to talk about stuff that I don't want your input on. So we're gonna go through a few stories that we've been thinking about in this reservoir viral immune environments and trying to put it together. And we're also going to talk about cancer a little bit. A lot of these patients are dying, persons are dying of cancer, and people with HIV have a higher risk of cancer, and we're trying to figure that out as well. This whole thing called a tumor microenvironment, and how is that related to the reservoir? So we can do a bunch of stuff. We can look at the tumor microenvironment, we can look at the non-tumor microenvironment, we can do our TCR sequencing, we can do our TCR specificity, and then we can guess of what is that receptor actually looking for. Is it tumor? Is it HIV? Is it CMV? Is it another virus? Is it bacteria? And then we can start to make these maps, and these are different last gift participants and what they look like in terms of their TCRs and where they're directed. So it gives us a good picture of what, one person is very directed towards other viruses, and while somebody else is very much directed towards a tumor, and then EBV and CMV are always high in the mix. And then we can also sequence the integration site of where HIV goes into that T cell. So we have a pipeline, and we have all the chromosomes, and we're looking for the HIV, and what gene is it going into in that T cell. I just want you to stop and think about that for a second. HIV is a retrovirus. It goes into our genes, right? And it disrupts when it goes in there. What is going on there, right? T cell now has a gene that is disrupted, or maybe multiple genes, and it's still alive. The T cell is still alive, right? So whatever it disrupted wasn't enough to kill that T cell. It was actually able to keep it going. But we can look at that integration site, and we can look at a bunch of T cells, and we can quantify that. And if they're all in the same place at the same time, then we can see that we'll call that clonal expansion. So one T cell gets infected in one gene, and then we see a bunch of T cells that look exactly that same integration site. We can call that a clonal expansion. And we can see where that gene is inserted. And then why did that gene allow that T cell to keep going? How come we still have clonal expansion of that T cell? And what does that mean in these microenvironments? And I'm going to talk about tumors, but we can also talk about microenvironments for HIV reservoirs as well. And this is just one participant, 74-year-old male, non-smoking, HIV diagnosis in year 2000, had lung cancer, a neuroendocrine cancer actually. It went to the brain, had a Mets. We can see all his different chemotherapies that he had, and then he had he died in 2022, and we did a rapid autopsy. And then we're going to be focused on this microenvironment, and we're just going to talk about tumor microenvironments now. But really, you got to think there's also an HIV reservoir microenvironment, and what all is going in that group. Well, there's tumor, of course, but there's also a ton of immune cells in there. And there's some of those are HIV infected immune cells. There's also bacteria in there, and nutrients and cytokines, and low oxygen is usually a characteristic of those tumor microenvironments. And then when we look at these tumor microenvironments, and this is anal cancer, breast cancer, and lung cancer, and these are the different groups where we might see the clonal expansion, and different tissues, so tumor, non-tumor, etc. We can see that there's quite a bit of difference in clonal expansion. That's all that I wanted you to get view here, is that clonal expansion is different for every different person in different tissues. It does turn out that overall cancer tumor microenvironments have a lot of T cells with HIV in them. So these tumor microenvironments have a lot of HIV infected T cells, and they tend to be very clonally expanded. And you can then look at where those genes are inserted and how those clonal expansions go. And in that far right one, that far one over there is anal cancer, and it's very much the HIV is inserted in a gene that disrupts apoptosis. Let's go back to what I asked that question before. What is that gene that is knocked out, and what is it doing to that T cell? Well, if it knocks out an apoptosis gene, then that T cell is going to survive. It doesn't apoptose, right? And in the middle one, it's an interferon stimulating gene. So it knocks out a gene that is associated with its responsiveness to interferon. So it doesn't do anything when its usual cognate chemokine, like, I don't care, I don't stimulate with interferon anymore. And then this one for lung cancer is P10, which is a very common oncogene. They call it oncogenes. I'm not sure I buy that term so much, but it is associated with lung cancer. So this T cell will not make P10. And we just got some really nice data yesterday showing that in that lung cancer tumor, we don't see P10 expression at the same places that we see P24 expression. So those T cells are not making P10, which is important to actually cause an immune reaction to the lung cancer that is there. There's a bunch of other genes. Like it gets crazy quickly mapping these pathways of all these genes that HIV has inserted itself into.
They're always inserted and inactivating the gene, or have you also seen hyperproduction of the gene?
We have yet to see hyperproduction of the gene. But what I think we will, if we keep looking far enough, because if it's upstream, we might be knocking out a repressor, right? Exactly.
And do you think that the infected T cells, so they get activated because of the tumor and then infected, or they get expanded because of the activation, or what is the, what
No
is?
idea. I think that once they get into this, and it could be a selection process. We make cancer cells every day, and our T cells and our immune system goes and clears them out. But if somebody has HIV and a tumor it starts its little process, and the T cell that goes to it is lazy and just doesn't react so well, it might allow that cancer to progress, and it's blocking other cells from getting there. And let's say it still is clonally active. And what I mean by that is it still has a signal to clonally proliferate. And we know it does because it's already clonally proliferated. And once it's in that tumor microenvironment, there's some sort of signal that tells it, okay, we need more of you. But the problem is it makes more of a crappy T cell. And that then makes that tumor microenvironment very conducive for cancer. This is all speculation, by the way. But this is something that we're working on. If you guys have any good ideas, love to hear it.
But are HIV infected people with HIV doing worse?
People with HIV do worse with cancer, period. There are AIDS-related cancers, so anal cancer, KS, but all cancers are worse in people who have HIV. And for a long time, people have said that's all inflammation related. They just have a higher systemic basal level inflammation, and we know that is associated with cancer. Okay, end of story. There's no smoking gun for that. But there actually might be a real HIV reason for people with HIV to have more cancer. Virologic reason. Yes.
For these participants, do you have you looked at the other immune subsets and see if there's
We are looking at other immune subsets. And Claudia's going to help us with some of those things, but also trying to sort out what is actually there. So what are the CD8s doing there, right? They're not infected with HIV. So are they actually helping or hurting, or are they blocked out? So in these tumor microenvironments, for a long time, we've known that they're inflammatory, but they're also hypoxic. So whatever's there is, it's hard, it's cold. It doesn't really do as much as it should. And if you have clonally expanding T cells that maybe is taking up more of that oxygen, it might actually be a problem. Yeah.
Do you see a differential distribution of those integration sites that keep coming up in the different tissues?
We haven't done enough. But what we are finding is that for the person's cancer, there's almost a few clonally expanded cells with HIV that have been associated with that cancer. P10 for lung cancer, that ND thing is for breast cancer, and then that PD2L ligand thing is for a lot of cancers, including anal squamous cell. The other one that's interesting is that some of these knock out chemotherapies. And what I mean by that is the immune checkpoint inhibitors need some of these genes to be active for it to regenerate these T cells, and they're now knocked out. Sorry.
Yeah, similar question. So the insertion sites, if you look at the distribution, how much are different pathways affected statistically? So are there some that happen more often than others, or is it more random?
It is, there is a pattern, and we don't have a full grasp on what that pattern is. But we are, and people have seen this for a long time. Since people started sequencing the integration sites, they said that HIV integrates into oncogenes. They said it from the very beginning. The problem with that is oncogenes are also very transcriptionally active, so they're open. So HIV goes into open chromatin. But that also, there is these connections. That's the reason they're called oncogenes, is that they are associated with various types of cancer. So sorting out that pattern, I'm probably going to need AI. or Antoine, who's going to figure this out for us, but we're just on this sort of story to figure out what the pattern is. Okay.
Do we know what's the function of this microRNA? What's gonna be there?
Yeah, so it's actually associated with interferon stimulation. So it is a microRNA, yeah. Okay, but complicated, right? That's a ton of data. The pattern is going to be difficult to figure out. Okay, I'm gonna stop there for a second. Any other questions? We're gonna change gears.
Question is with the co-infections, you showed us this pie graph where there was a lot of viral load on others. Is there a preference for certain other viruses, hepatitis B, hepatitis C, or is it just random or across the board, or could there be some synergistic effects?
I'm sure there's synergistic effects, and if Sarah Gianella was giving this talk, she would talk a lot on the herpes viruses because the T cells are directed a lot towards herpes virus. CMV and EBV drive a lot of these T cells. And of those that are clonally expanded, a lot of them, if not most, are CMV directed.
Thank you.
Okay. We'll start back to that question that I asked at the beginning. Would you be interested in participating in a study at the end of your life? That's a big ethical question, right? So one of the big things that we've been doing in this study is having a big ethical component. And this is work from Corinne and Davey, with lots of interviews and lots of studying and asking next of kin and asking them how they feel. One thing that we're just now seeing a signal for, which I think is fascinating, and I'm hoping to turn it into a randomized controlled trial, is that people who participate in the study have a better quality of life. I think I put it on there, than people who don't end up being on the study. Why do you think that might be?
Give them a purpose.
Yeah, that's exactly what they say. I now have meaning in the last bit of my life. It doesn't feel wasted anymore. So they have a better feeling about their death, which is amazing for me. My death will not be in vain. That was something we hear over and over. So the ethics of it might be this is something we should be doing to help people have meaning at the end of their life. But then we asked them another question. Actually, we asked them the question at the very beginning. So all those other ones that I asked them another question. Would you be willing to be exposed to an infectious pathogen at the end of your life to do research? I gave them streptococcus, so strep throat, HIV, hepatitis, and malaria. So some of these people didn't have HIV, remember? And boy, 40% of people said they were willing to do at least one of those, right? And the most common one was a third was strep throat. And strep throat is the worst one on there as an infectious disease talk. I mean, it's horrible, but they knew about it, right? They probably had it sometime in their life, and they're willing to do it again. The other ones were just more scary, like malaria or hepatitis C or HIV, right? But still, almost a third of people would be willing to be exposed to HIV at the end of their life to participate in a research study. This is crazy, right? But let's just think about it for a second. If I had 12 people and I had a vaccine, right, an HIV vaccine, and I gave it to 12 immunocompetent people who are in their life dying of, let's say, brain cancer or ALS or something, I randomized it, I exposed them to an exact dose of HIV, I would tell you which vaccine would work and which vaccine wouldn't work pretty quickly versus 1,000 other people who are otherwise healthy. Same thing for malaria, same thing for strep throat, same thing for PrEP, same thing for any of the prevention studies. Okay, so we started this conversation, and boy, so we got this one paper out, but everybody, the community advisory boards are now really pushing us like, this is the way that we want to go. Right? And you can think of, boy, non-human primate studies, those sorts of things. We might not need those as much anymore. And this has been a big push from our community advisory board. Okay. That's a big bucket. So we're thinking about interventions. We've had conversations with the FDA already. The FDA at the beginning has said, can't do that, that's just nuts. And then we just asked them the question, would you be willing to do it? 90% of them say, yeah, but I'm different. That's always what we hear. I'm a scientist. I'm logical. I believe in science. I'm like, yeah, so do other people. And there's a risk-benefit ratio that you're supposed to consider. And once that conversation started happening, they consider the last gift cohort having a different risk-benefit ratio versus someone who has HIV and is otherwise healthy, or anybody else that would go into a phase 1 study. So they've given us this sort of goal to think about phase 1 studies that have a different risk-benefit ratio than you would consider for something else. And for looking at studies that you can't do in otherwise healthy people. So one of the ones that we just sent in for an ACTG study, AIDS Clinical Trial Group study, completely boring, but very interesting in another way. So VCO7 or VRCO1 is a neutralizing antibody. It's been around forever. It's completely safe, but I don't know where it goes. So to Melanie's question, does it actually get in the brain? I don't know. And they're now making a long version, acting version of it. So they want to give it to somebody, and so they don't have to take medicine for 6 months. Does it get in the brain? I don't know, but I can tell you that we can give it to some people, and then we can go look in the brain after they pass away. Perfectly safe, perfectly good intervention. It's an open question that we could actually test in this group. And then you're like, why can't I do that in non-human primate? Well, some of it I can, but their reactivity to these humanized immunoglobulins are just different. So it will really need to be in a person. Other ones, CAR T cell projects. I just had a conversation with Steve Deeks. He has a CAR T program. I have a CAR T program for HIV cure. But wouldn't you like to know where those CAR Ts go once you give it to somebody? Do they actually get in the brain, kidney, lymph nodes, spleen? Is it all the same? Is it different places? Are they going to the reservoirs? This could be a place that we could look at, find it out. Noise enhancers, LRAs, drug penetrations, in vivo imaging would be really nice. So now there's all these studies looking at PET. Can we characterize the reservoir with these immune markers, imaging markers, etc.? But let's do it. And then at the end, when we have a rapid autopsy, go back and look in those tissues and see whether or not what you're seeing in the imaging correlates with what you actually see in the tissue. And then vaccines, inflammasome drugs. And then what ideas might you guys have? So we're interested. Okay, let's do it. I am all on board. Okay. Some acknowledgements, tons of people to do just a few autopsies a year. We actually are now funded for 1 year to enroll 5 people who don't have HIV. So there are controls. And they are just as willing to participate in these studies in people who have HIV. I thought that maybe they wouldn't be, because people who have HIV have lived through HIV and oftentimes were participating in research, and they were very focused on that. But boy, when somebody's like, I want to cure a disease, they're like, sign me up. And they're oftentimes get asked, like, I have diabetes, I have people who in my family have diabetes, are you going to do diabetes research? So it's just human nature to think about that altruism, and it really does, give me hope for the future. And these are our collaborators across the US. Happy to add more dots on there, because there's one of our big things that we've been really interested in is to make sure that the people who give us this special gift really do get the most science out of it. So we've been at the very beginning, like, just pushing as many tissues and data as much as possible out to people who can actually do something with it. So if you have ideas, let me know. All right. Anything else?
I don't know if you have enough numbers for this, but do you see sex differences in distribution in the tissues or the evolution of the virus or anything like that?
Well, we don't have a lot of numbers, but we don't see much difference at the moment. There's a caveat to that. I do believe the estrogen receptor story around differences in the reservoir. Most of the women that we have in this study are postmenopausal. So they've probably been not under the influence of estrogen the same as in other places. So we haven't seen that too much.
Do you have enough N for your treatment studies, for example, on how much?
We would have to enroll more. So I would need, I need a new grant. It would be, and it would have to fund exactly the intervention off that new grant.
You only enroll in San Diego, or do you go also to different sites across the country?
Only in San Diego, because that's where the team can actually do it. So there's two parts to that. One, we got plenty of people in San Diego. We get to be a little choosy just because there's not that much money. These are super expensive. But we are extending into Palm Springs. So there's a very strong community group there who's very interested in the study. And we've done one participant where we're able to, after they pass away, put them in an ambulance and get them down relatively quickly. So I think that's going to be doable.
The HIV challenge studies that you mentioned that people without HIV are willing to do you see that happening in the next 5 years or 10 years? Do you think that could be implemented?
I think so. I think there's two reasons that could be. One, they did it during COVID. And two, once we show that this is an acceptable intervention and something that I think is going to be rather easy and safe, I think that the FDA will have a less risk attached to this, and they should consider these HIV challenge studies. I think it would revolutionize how we do our vaccine studies. Okay, yeah.
Yeah, for some of your control participants, the HIV uninfected but not negative, do you have any of those participants that have some sort of
Malignancies? Yep. So we're matching them to the malignancies that the persons with HIV have. So that's actually that's what we're trying to do, is to do some matching on that. Age, gender, and then comorbidity as well. But they're not perfect and they're small numbers. Yeah, sorry, go ahead.
I think the challenge studies are not going to be very useful for vaccines, just because vaccines don't work very well in old people. I mean, you're just the age group, so that's a caveat.
Yes, and we have people who are 52, but we do have people who are 50 and less in our study, especially on the ALS side. So the ovarian and brain tumors. So yeah, older people die, but young people die too. And it turns out that the younger that somebody is, the more they want to get back. It is really compelling. So I don't think it's going to be a problem. If I put a shingle out and said, you have 6 months to die, would you be interested in a vaccine study? I would have a queue lined up to do that study.
So when you started this was really sort of avant-garde. Is it still avant-garde, or are there people doing the same thing in terms of, because actually I think you pointed out it would be something that every disease could benefit from, and do you see more studies like yours coming up in other areas, or what's the environment?
People are interested, and some of it is starting to catch on. Like we have a meeting when I get back with a group in Brazil who want to do this, which is nice. And there was one that started in South Africa. They can't quite get the rapid part of the autopsy together. There are a lot of people who want to do it. What is interesting though is the reinvigoration of the other diseases. So like cancer, pretty much had stopped their rapid autopsies for a long time. And then we started talking about it and they kind of opened back up and started doing these again. And the same thing for ALS and Alzheimer's. So Alzheimer's also has they do tons of autopsies, but they've never really done the rapid autopsy. So now we're talking about so they're using basically our SOPs to go in to do these rapid autopsies for the Alzheimer's
Because there's a lot of brain tissue that is being banked from the
That's
autopsies,
right.
that it would benefit from a rapid protocol.
Yeah, if you go in and you look at those tissues from the Alzheimer's after 12, 24, and sometimes 48 hours after somebody died, you're not going to get any RNA. The protein is completely degraded. You can't figure out what is activated and not activated. It's just, yes. So really trying to push to this rapid side, I think is going to be the key, especially for the newer technologies. CyTOF, single cell RNA-seq, you really gotta get in there fast.
Sort of circle back to a previous point you touched on. Do you think that like with the development of traditional treatments like or treatment and like because I know that there are injectables right now like twice a year. Although it does not reduce the RNA load in the brain, but do you think that will, with that development ongoing, do you think that will eventually undermine the importance of finding a cure for HIV?
No. Every single person that I ask who has HIV, are you interested in a cure? Tell me yes. Right? It is, and there's been a lot of economics around this. Economists for some reason are really interested in this question, and they view it as around $1 million thing. Somebody's willing to come up with $1 million to be cured of their HIV. Even though taking a shot twice a year, or maybe soon once a year, it's important to them. And it's more emotional or stigma or those sorts of things that are attached to it.
And it's not solving all the problems, because that's the problem, no? I mean, there's still chronic inflammation, transcription of the active reservoir.
Cancer.
Cancer. I think the push to make the last hurdle, because it's not that it is solved.
Yeah. The people who have HIV, when I see a patient and they're devastated after I have to give them their news, I tell them, take your medicine, you're gonna live a long, happy life. And that is that one person. But if you look at the population as a whole, they still have a less lifespan, and they have more chronic morbidities. So I like to get rid of that. Good question though. Other thoughts? Awesome. Well, thank you for inviting me. This is super fun. But there are people online. Is there anybody wanting to talk about it?
It's the next thing.
Hi,
Yeah.
Hi, Dr. Smith.
Hey, just wanted to make sure I saw you. Hope you're doing well.
Just wanted to say hi.
Back at you. That was a great talk. Thank you.
Yeah,
With the trainees, exactly. Very thought provoking as always, and yeah,
Super fun.
would love to talk about a little few things about that. Okay,
All on
see
board.
you at 1.
Nice. So we'll go to that.
Okay. So I'll follow you. The plan. You'll take me?
Yeah, yeah.
Okay.
We'll work.
I think it's good that you asked. There are some yes, there is. I don't know if you know the word list. If you avoid putting any word into the list 60 from that list, you know, okay, we'll talk about that. Cool. Okay.
Question.
Yeah.
Yeah.
I don't know if you remember me, but I used to work with Davey.
Oh, yeah. Yeah, you're here. How's it going?
That's great.
Yeah.
I'm working JAMA right now.
Oh, right.
It's like when I worked there, it's a good first experience. For all of this stuff.
Yeah.
Now I'm applying PhD.
Lizzie now on the lab.
Yeah.
Interrupt.
Yeah, I was focusing on other things, but at least for in your data set, it doesn't look like in this lines. It's a difference.
And I find your presentation really intriguing. So I'm already in the neuroscience department, but during college I participated in the we're a local pharmaceutical company trying to develop their HIV as well. So we did like a health economics research.
Yeah.
So yeah, I just find everything very interesting. And also I kind of just maybe a quick question.
Sure.
How do you envision because right now there is a severe uneven distribution of CAR T-cell facilities, facilities that are capable of doing CAR T-cell therapies across the US and also across the globe. So how do you think that will contribute or prevent the
I
stuff?
think if we get a CAR T cell that it cures HIV, then more will be made, and hopefully will be more efficient at being made. So we see that over and over and over again, like monoclonal antibodies for COVID.
Mhm.
Only a few places were able to do it. As soon as they were made, boy, then the production really shot up. So that's one. And two, CAR Ts, I think, are going to be interesting because we're going to learn something, but is it going to be broadly available to everybody in the world? Probably not. But what we learn might get us to the point where something will be broadly available.
Yeah, sounds good. Thank you so much.
Yeah, thank
This is
you.
really interesting.
I'm glad it was interesting.
Uh,